Good morning, students. Welcome back to Botany class, Chapter 2, Biological Classification, Part 1. Okay, last chapter we have studied how to identify the organisms, how to give the names, taxonomy, identification, classification, and naming of an organisms. Now, here we are again giving to the how to classify the organisms. Only headings we have studied last chapter. Now that is how to classify the living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, like for food, shelter, and clothing. Without any type of the criteria, we are classifying the living organisms. The scientist, father of the biology, Aristotle, was the earliest to attempt a more scientific basis for classification. He used simple morphological characters. What is the meaning of morphology? The external characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. After that, Carlos Linnaeus he divided the classification into two kingdom classification there are only two kingdom classification. First one, it is kingdom Plantae and kingdom Animalia. That is Carlos Linnaeus. And kingdoms was developed that include all plants and animals in that group. This system was used till very recently. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. A need was also felt for including, besides gross morphology, other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Okay. After that, they have started the classifying the organisms depending on the cell structure, nature of the wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over time, though plant and animal kingdoms have been a constant under all different systems. The understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over time. Later, R.H. Viktagger, the name is very important, R.H. Viktagger in 1969 proposed a 5-kingdom classification. Okay, R.H. Viktagger in 1969 proposed a 5-kingdom classification. The kingdoms defined by him were named Kingdom Monera, Kingdom Protista, Kingdom Fungi, Kingdom Plantae, and Kingdom Animalia. The main criteria for classification used by R.H. Viktagger include cell structure, thallus organization, mode of nutrition, reproduction, and phylogenetic relationships. Gives a comparative account of different characteristics of the 5 kingdoms. Okay, R.H. Viktagger based on the cell structure, thallus organization. Thallus means the plant body is not divided into the root, stem, and leaves. according to that level, mode of nutrition, reproduction, and phylogenetic relationships. Let us look at this 5 kingdom classification to understand the issues and considerations that influence the classification system. Earlier, classification systems include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unfitted those whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group. What is the meaning of heterotrophic? Animals are depending on other organisms for food. Example, the fungi and the autotrophic green plants. What is the meaning of autotrophic? They are preparing their own food, green color plants. Though they also showed a characteristic difference in their wall's composition. The fungi had chitin in their cell wall, while the green plants had cellulose in their cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom that is called kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, it's a type of algae. They have the cell walls with the paramecium and amoeba, which were placed in the animal kingdom because they lack the cell wall. They are the absence of the cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve classification system which reflects not only the morphological, physiological and reproductive similarities but also phylogenetic, means based on the evolutionary relationships. Now we will discuss about the characteristics of kingdom Monera. Okay, the first kingdom, R.H. Viktagger, based on the classification, the first classification was kingdom Monera. Bacteria are the sole members, that are the basic members of the kingdom Monera. The members of the kingdom Monera are known as the Monerians. They are the most abundant microorganisms. Lot of organisms are present. Bacteria occur almost everywhere. Hundreds of bacteria are present in the handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans where few other life forms can survive. Many of them live in or on their other organisms as a parasites. Okay, bacteria can live in any other extreme conditions, extraordinary conditions, very hot, very acidic or very hot desert or very ocean. There also the bacteria are present because they can survive with their cell wall. Okay, otherwise if they can live as another organism, live in another organisms as a parasite. Then the bacteria are grouped under 4 categories based on their shape. Okay, shape of the bacteria. They are divided into 4 types. The first one, spherical bacteria. Spherical means round-shaped bacteria are known as coccus. Round-shaped spherical bacteria are known as coccus. The rod-shaped bacteria are known as the bacillus. Rod-shaped bacteria, bacillus. Comma-shaped bacteria are known as the vibrium. Comma-shaped vibrium. And the spiral, spring-like structure bacteria are known as the spirillum. Though the bacteria structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show that the most extensive metabolic diversity. Some of the bacteria are autotrophic, they will prepare their own food, they synthesize their own food from inorganic substrates. They may be photosynthetic, autotrophic, or chemosynthetic autotrophic. They are depending on light, otherwise they are depending on other organisms' food, that is called chemosynthetic. The vast majority of bacteria are heterotrophs. They do not synthesize their own food but depend on other organisms or dead organic matter for food. They are known as the heterotrophs. The bacteria are divided mainly into two groups, Archaebacteria and Eubacteria. First we will go for the Archaebacteria. These bacteria are special since they live in some of the most harsh habitat where they are living, very harsh habitat such as extreme salty area. Archaebacteria living in the salty areas, bacteria are known as halophiles, HALO, halophiles. Archaebacteria living in the hot springs are known as the thermoacidophiles, and the Archaebacteria living in the marshy areas, methane gases, they are known as the methanogens. Archaebacteria differ from other bacteria, normal bacteria, having a different cell wall structure, very important cell wall structure, and this feature is responsible for their survival in the extreme conditions. Because of the bacteria's cell wall is helping to survive in the extreme conditions. Next, methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane. It's a biogas from the dung of these animals. Methanogens are present inside the animals, cows and buffaloes, gut of the animals. Next group of bacteria are known as the Eubacteria. There are thousands of different Eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall. Their rigid cell wall is present, and if motile, if they have the movement, the flagellum is the locomotory structure for the bacteria. The cyanobacteria, also known as the blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. With the help of sunlight, they are synthesizing their own food, cyanobacteria. The cyanobacteria are unicellular, one cell, colonial, they are present like in a colony-like structure, or filamentous. They are the filament-like structure in the colony, or one cell, or in a colony, or it is in a filament-like structure. Freshwater marine or terrestrial algae. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocyst. What is the function of heterocyst? They can fix the atmospheric nitrogen in the specialized cells are called heterocyst. Examples: nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various organic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. Energy currency of the cell is the ATP, adenosine triphosphate. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Chemosynthetic autotrophic bacteria plays a very good role in the atmosphere. Then next group of bacteria, heterotrophic bacteria, are the most abundant in nature. The majority are important decomposers. They are helping to the decomposition. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotic, fixing nitrogen in leguminous roots. What are the importance of the bacteria? How it is helping to the human beings? They are the decomposers and they are the helpful in making curd from milk, production of antibiotic, fixing nitrogen in the leguminous plants, rooted plants. Some are pathogens, disease causing. They are causing damage to the human beings. Not all the bacteria, some of them are harmful. They are damage to human beings, crops, farm animals, and pets. Some of the diseases: cholera, typhoid, tetanus, citrus canker are well-known disease caused by different bacteria. Okay. Bacteria reproduce mainly by fission, binary fission, dividing into two. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. Next important point, one more type of the bacteria in the kingdom Monera, in that the mycoplasma are the organisms that completely lack cell wall. Cell wall is absent in mycoplasma. They are the smallest living cells known and can survive without oxygen. Okay, mycoplasma do not have the cell wall. They are the smallest living organism and they can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Thank you.